I didn't have so many board things. I actually did something to my house last night. I dropped my signature and texting right now. And then he was like, she had her time and it's really good thing. And I told her, like, first day sleeping in 10 days. I feel like I need more.
Okay, if you're looking at what's up on the screen right now, this is where we left off last time. We're dealing with important groups of gram-positive bacteria. So we're still in the phylum Firmicutes. Most of these are ordinary gram-positives. However, some things that used to be categorized as ordinary gram-positives have been moved out of this phylum. Anyway, we looked at Bacillus already, so we're moving on. So the next important phylum is Staphylococcus. We've worked with Staphylococcus a little bit because they were on our mixed egg plates. We have Staphylococcus aureus. These organisms have some important characteristics. One that we typically test for in here is catalase. If you recall, catalase enzymes associated with peroxisomes break down hydrogen peroxide. Bacteria don't have peroxisomes, but they still produce catalase enzymes, and they do the same thing. They break down hydrogen peroxide. So to test Staphylococcus for catalase enzyme production, you expose them to a little bit of 3% hydrogen peroxide, and if they bubble, they're catalase positive. It's a really easy test to do, and we do it frequently. Beyond that, the name Staphylo means they are grape-like clusters, and coccus means they are coxy or spherical-shaped cells. So their name tells you what they look like. Sometimes they form carotenoid pigments, sometimes they don't. Carotenoid pigments, look at the word parrot, think orange, yellow, that's carotenoid. The staph that we work with is white, so they're not producing that pigment, but some staph do. So there you can see some Staphylococcus colonies on a plate. That particular plate, the colonies look yellow, but that's because that's mannitol salt agar, and there's a pH indicator in there that's causing yellow color. So we know that Staphylococcus aureus is potentially an important human pathogen. So what did we have in the very first week of outbreak? No, it was the second week of outbreak. We had toxic shock syndrome. Staphylococcus can get in the bloodstream. They can cause septicemia. They can cause toxic shock syndrome. They also cause a number of skin infections. They are often associated with what are called nosocomial infections. These are institutionally acquired. So people going into a clinical setting, maybe they go in without the Staphylococcus and they pick it up there, which is not okay. And then, of course, people are very concerned about MRSA. One of those acronyms that people try to pronounce as a word, which drives me nuts. MRSA, which is MRSA. The SA is Staphylococcus aureus, and the MR is either methicillin resistant or multiply resistant. Those are organisms that are resistant to at least methicillin and sometimes a wide range of antibiotics, and they are quite problematic. So there we see some colonies of Staphylococcus, skin infection, and an infection in the foot involving Staphylococcus. Okay, the next genus is Lactobacillus. This one, the bacillus in the name tells you that they're going to be rod-shaped organisms. The lacto in the beginning has to do with these guys producing lactic acid. So these are gram-negative bacteria, but they are among multiple organisms that are sometimes referred to as lactic acid bacteria because they produce lactic acid as a waste product of the fermentation activity. It just happens that lactic acid is something that we as humans have acquired a taste for. So if you have sourdough bread or sourdough pancakes or something along that line, it's likely that the material that they're made out of is likely to contain lactobacillus. So lactobacillus species are used extensively for making cheese, yogurt, sauerkraut, sourdough bread, etc. They're not the only lactic acid bacteria, however. This next genus, Lactococcus, these organisms were originally categorized as Streptococcus, and there are several different species of Streptococcus that are also lactic acid bacteria. Once people started looking at bacteria and using 16S ribosomal RNA nucleotide sequences to identify, they said these guys are not Streptococcus. And so a new genus was established. The genus is Lactococcus. They are like Streptococcus in that they are catalase negative. They haven't gotten to the Streptococcus yet. So Staphylococcus are catalase positive, the Lactococcus are catalase negative, and it's a pretty easy test to do. These guys are also used in the production of cheese. In fact, they're probably the most common. We have a chart of different types of cheeses that it's in the lab manual, so when you get to the food microbiology section, you look at a lot of different types of cheese and they will tell you that the cheese contains Lactococcus, lactis, or cremoris. Well, it turns out they're all Lactococcus lactis, and then lactis and cremoris are subspecies. So we've talked about the binomial name, where you have a genus name followed by a specific epithet. Sometimes there's another name following that, which is called the subspecies. So we have Lactococcus lactis lactis and Lactococcus lactis cremoris. It's probably more about those guys than you wanted to know. Anyway, they do produce lactic acid as a byproduct, and that's what makes the sour flavor in cheese, lactic acid. Then that brings us to the genus Streptococcus. Once again, this genus name tells you something about the organisms. Cocci means the shape, and strepto means they are in chains. So we have cells arranged in chains. They are catalase negative. They are homofermentative, which means they only produce lactic acid as their fermentation products. If they're involved in food production, then they're producing lactic acid. But that's not the only thing they do. So there are clinically significant Streptococcus. Streptococcus pyogenes causes strep throat, scarlet fever, rheumatic fever. Streptococcus pneumoniae causes pneumonia. It's usually the most common cause of pneumonia in humans, although Neisseria meningitis can also cause pneumonia. And a bunch of other things. This business of being cocci in chains, you'll typically see this kind of arrangement if the organisms are grown in raw. And they really are cocci, but they tend to elongate when they're getting ready to undergo fission. So very often they'll look like short rods. This is a picture of strep throat, so the throat is very red with Streptococcus pyogenes. And if you've heard of the flesh-eating strep, the flesh-eating strep is a particular strain of Streptococcus pyogenes. Not the strain we have in here, by the way. Streptococcus thermophilus, these are organisms that are used extensively in food production. So although some Streptococcus are pathogens, not all Streptococcus are. Streptococcus thermophilus is used in the production of string cheese and yogurt. So again, lactic acid bacteria. So this brings us to our next phylum. Again, ignore the phylum numbers. We're coming to the phylum Actinobacteria. These bacteria occur very commonly on air plates. So if you look at this side of the display case set over here, a whole series of these organism types that are in the phylum Actinobacteria. So starting here, we have Arthobacter, Procuria, Micrococcus, Dermacoccus, etc. There's a whole bunch of these guys that are categorized as Actinobacteria. For a long time, people thought Micrococcus and Staphylococcus were closely related. Turns out they weren't. So now the Micrococcus are in a completely different phylum. So Micrococcus, this was a genus that...
Yes, sorry, before we move on to that one, what was the last phylum that we're talking about?
We were doing Firmicutes before we moved into Actinobacteria. Okay. So Micrococcus, in the Burgess manuals that were published in 1989, all the gram-positive cocci were put together in a kind of a cluster, and they were supposed to belong to the family Micrococcaceae, as though they were all related. They had Deinococcus in there, Staphylococcus in there, Micrococcus in there. Well, it turned out those organisms weren't closely related at all. And so they kind of pulled them out. The Micrococcus stayed in the phylum Actinobacteria. These are catalase positive, they're gram positive, they're coxy. They're extremely common on skin of humans and other animals, and so they tend to be associated with soil and sand and whatnot. We run into a lot of them on our air plates, and they're interesting because they're brightly colored. So some are yellow, some are pink, some are kind of orangey. Well, there were quite a number of these organisms that were all in this one genus until people started looking at them more closely, and then they found out, they're not really all Micrococcus. So they're still all actinobacteria, but now this genus Micrococcus has been split into Archobacter, Cochuria, Paedococcus, Dermacoccus, etc., etc. Nevertheless, if you decide to use your air plates as a source of semester projects, if you pick colorful colonies, it's quite likely that you'll encounter something, one of these. It might be Micrococcus, it might be one of these others. And this name, Dermacoccus, kind of tells you where these things live. They live on skin. So we see a lot of these. Again, they're kind of fun to work with. They're not usually pathogenic, so they're not generally considered to be pathogenic. They can infect people that are immunocompromised, but they're brightly colored. They're pretty easy to grow, pretty easy to work with. So we see a lot of them. You see pink ones, yellow ones, kind of orangey ones. The next phylum is the Corynebacterium phylum. These things for a long time were just referred to as coryneiforms, and in clinical settings, oftentimes people couldn't identify them accurately, and they'd just say, we saw something under a microscope, it looked like some kind of coryneiform. These produce bacilli that are not uniform bacilli. They tend to look like clubs, so they're kind of narrow at one end and fatter at the other end. In this phylum, we have Corynebacterium, which is actually a genus. We're in the phylum Actinobacteria. And again, club-shaped. Oftentimes these things form what's called a snapping arrangement, where you'll see them in this type of a shape. I saw a movie recently that showed how these organisms undergo fission. So if you have one rod-shaped cell, they undergo fission, they actually snap apart. So something happens to their wall, and the two pieces come apart, but they don't separate totally, so they're hinged at the corner, and they form these odd snapping arrangements or V-shaped arrangements. One is a very important human pathogen, Corynebacterium diphtheriae. These cause diphtheria. Not usually common in this country because most people are immunized against diphtheria. We hope most people are. There's one called Corynebacterium pseudotuberculosis. This one causes a disease in horses called pigeon fever. I don't know why it's called pigeon fever, but it causes the development of these abscesses just under the surface of the skin. Oftentimes these abscesses occur where the animals have injured themselves. The Corynebacterium live in the soil, and if they get up into that wound, then there's a lot of pus produced underneath, and you'll see this drainage coming out of the wound. This stuff will fall back onto the soil, and then you have more Corynebacterium in the soil. So it's pretty nasty. Apparently, there is a vaccine that has been recently developed to control this because it's quite problematic otherwise. There are lots of other Corynebacterium. Then we come to the next genus, Mycobacterium. What do we know about the genus Mycobacterium? Anything? Do you remember what we did with them? They were on plates. Yes, they were on plates. They looked like this.
Acid fasting.
Acid fast, right? Mycobacterium, they're acid fast. Okay, so they are acid fast, and the reason they are is because they have high levels of mycolic acid in their cell wall. This makes them resistant to staining with a graph stain. If you heat them and use a lot of time with a very potent stain, you can get them to pick up the stain. So they are acid fast. Again, in this category, there are some important human pathogens. Mycobacterium leprae that causes leprosy. Mycobacterium tuberculosis that causes tuberculosis. But there are many, many types of Mycobacterium that live commonly in the environment and are not generally considered to be pathogenic. The other thing that's kind of interesting about the Mycobacteria is they tend to be very slow growing. And when they produce colonies, the colonies are kind of waxy because of the mycolic acid, crumbly when you touch them with a loupe, uncooperative when you try to make smears. Isn't that right?
Yeah.
The next genus is Nocardia. This is one we don't have an example of. We don't have any representatives of Nocardia in our morphological unknown set, unfortunately. Probably the biggest thing about these is that they make cells that are kind of connected together in chains. So they have these things that are called, well, they look like hyphae, which is a term that relates to fungi. We'll get to that in a bit. Oftentimes these chains will break and you'll have cells that are Y-shaped. So they'd be like this, with two prongs, which is very unusual for bacteria. Some of them are opportunistic pathogens. So Nocardia asteroides, which is shown here, is an opportunistic pathogen. And then we come to the genus Streptomyces. These are truly filamentous bacteria. So when we talk about bacterial shapes, you have cocci, you have bacilli, you have spirilla. These are none of those. The Nocardia were kind of leaning toward making filaments, but the Streptomyces are definitely filamentous. So they form these long strings, and off the ends of these filaments, they form conidia, or conidiospores, so that when you look at the surface of the colonies, they look powdery. These are the conidia in an electron photomicrograph, and here's the surface of the colony. They are maybe smooth initially, but then they'll begin to develop this powdery surface, and it's all those conidia. These things will actually dump the conidia out just beyond the border of the colony, and then they'll start a new area of growth. And so oftentimes when you look at Streptomyces colonies, you'll see layers of growth like concentric rings. One of the important features of Streptomyces, these are an important source of new antibiotics. So there's a lot of different antibiotics that originally were obtained from Streptomyces. I have a list there: tetracycline, streptomycin, neomycin, erythromycin, vancomycin. If you look up vancomycin now, what you'll find out is the organism that produces vancomycin is not Streptomyces. It used to be, but the name has been changed, and that's happening a lot with taxonomy. So they are an important source of antimicrobial drugs. Another interesting feature of Streptomyces, the cultures often smell like dirt, or we think of them as smelling like dirt. So if you have Streptomyces, if you open the plate a little bit, you might get the odor of fresh soil. Well, it turns out Streptomyces don't smell like soil. Soil smells like Streptomyces. So Streptomyces produce a substance called geosmin, and it is geosmin that makes soil smell the way it does. If you've ever had fish that were caught and maybe they were living in a pond, and if you eat the fish, it tastes a little bit like dirt, that's geosmin. And oftentimes that's not from Streptomyces, it's from cyanobacteria, because they also produce geosmin. I was trying to think, who has the Streptomyces? Oh, you have the Streptomyces, because you have to keep putting it in the broth. Do they smell? You have to smell them?
I have it on my
Why don't I pull them out and take a sniff of them? Okay, moving to the next phylum, the phylum Chlamydiae. Notice the spelling at the end, IAE. The genus that's important is Chlamydia, so it's still spelled the same but missing the E on the end. Anyway, these are gram-negative. They don't have peptidoglycan when they develop inside a host, so these are pathogens. And within the host, they form these little bodies called reticulate bodies that don't have peptidoglycan. The other feature of chlamydia, for a long time researchers thought these things couldn't produce ATP. Well, if a cell can't produce ATP, it really can't carry out any physiological processes. That's their power supply. So it turns out they can produce just a little bit of ATP, but they're very much dependent upon their host. So they are hypotrophs. They're obligate intracellular parasites. They reproduce inside some other organism or inside another cell. And they don't require vectors for transmission, which a lot of hypotrophs do. So what's the most common thing that we know about chlamydia?
STD.
You know? And it's a very common sexually transmitted disease type organism. That is, chlamydia trachomatis causes sexually transmitted disease. So it can also get into the eyes and cause blindness. this is a very serious problem. If a mother is infected with chlamydia, when an infant is born, the infant can develop eye infection with chlamydia. And then the phylum Spirochetes. Do you remember anything significant about spirochetes? When I was talking about prokaryotic cell structures, there is a periplasmic space, and only the spirochetes have a flagellum in that space. So these are very thin, they're gram-negative, but they're very thin, spiral-shaped bacteria, and they have periplasmic flagellum. They're really difficult to see with a light microscope. They have to be stained especially to see them. So here you can see some images of spirochetes. Little skinny spirals, some loose spirals, some tighter spirals. And there are some significant pathogens in this group. So Treponema pallida, causative agent of syphilis, Borrelia, there's Borrelia burgdorferi, and now there have been other species of Borrelia that have been discovered that cause Lyme disease, and Leptospira. Some of these are tick transmitted, but I don't believe all of them are. I don't think Leptospira has to be vector transmitted. Now, I just stuck this slide at the end because this seems like a lot of bacteria, and yet we know maybe maximum 10%. That is, we've been able to culture maybe 10% of the bacteria that are on the planet. So we really don't know much. So, trying to kill off 99.9% of them, probably not real smart. We don't know what they're for. We don't know what they're doing. We don't know whether they're beneficial. We have this assumption that the only good bacteria are dead, and that is far, far from the truth. So if you are using a lot of antibacterial products in your home, stop it. It's not a good idea. Alrighty. From there, we're going to go to fungi. So I wore this shirt that says I'm only 10% human. If you remember the human microbiome thing that you went to doing your web-based lab assignment, and it said that we're maybe 25% humans, or 25% human cells. I come to this conclusion. It's somewhere between 10 and 25%, depending on how many bacteria are present in your gut. And the gut contents changes significantly depending on what's going on. Have you eaten recently? In which case you have a whole bunch of more bacteria than you would otherwise. Just had colonoscopy, for example. The percentage of bacterial cells goes way down. But anyway, most of the organisms that live with us are bacteria and fungi. So we're going to look at fungi next. Very important jump here. When we were talking about bacteria and archaea, those were prokaryotic cells. Now we're moving into the domain eukarya. And all the other groups we look at are in the domain eukarya. This is a definition of fungi that I took from a mycology textbook. Mycology is the science or study of fungi. It says fungi are nucleated, achlorophyllous, osmotropic spore-bearing organisms that typically reproduce sexually and asexually, and whose usually branching filamentous bodies are surrounded by cell walls composed of cellulose, chitin, or both. Like, well, that's handy. Well, it tells you these are organisms that have cell walls. That was probably one of the criteria that caused people to initially group them as plants. I'm going to try to convince you they are not. Fungi are not plants. The singular is fungus, plural is fungi. So there's some kind of joke about some dude goes into a bar and he's trying to talk to these people and nobody will have anything to do with him, and after a while he says to the bartender, I just don't understand this. I'm a fungi. He couldn't be. There's no such thing as a fungi. Fungi is plural. He had to be a fungus. Bad joke. Okay, so again, the cells are nucleated, which means eukaryotic, right? Eukaryotic organisms are truly nucleated or well nucleated. So They are eukaryotic type cells. And achlorophyllus, we know that A or N in floatable words means without. So what are they without? They're without chlorophyll. What's the significance of chlorophyll? Anybody remember? Chlorophyll is associated with organelles in eukaryotic cells that are called chloroplasts. The chlorophyll pigments are part of the inner folded membrane, the thylakoids. Chlorophyll molecules are light trapping. So they capture light energy and they allow plants to generate ATP using light. Fungi can't do that because fungi don't have chlorophyll pigments. They're achlorophyllous. So these things are not photoautotrophs, which plants are. Instead, they're chemoheterotrophs. They're like us. We're chemoheterotrophs. So we take in organic compounds and break them down, and we get our energy from organic compounds. Again, not plants. Under the heading of chemoheterotroph, we had lots of subcategories. One of the subcategories was saprotroph. Saprotrophs are organisms that use primarily dead and decaying material. Well, the reason the materials are decaying is because the fungi are breaking them down. What fungi do is dump a lot of digestive enzymes into their environment. Through exocytosis, they put those enzymes out there. This allows them to break down the food outside and then take in nutrients in a smaller form, take them in as molecules. The term that was in the definition, osmotrophic, I don't like that term because osmosis is the movement of water, and osmotrophic has to do with fungi taking in organic compounds by absorbing them. They're not really, they take them in through endocytosis. So I'm not going to ask you the term osmotrophic, I don't like it. Makes no sense. But fungi are not plants, even though when you look at them, they have kind of a plant-like appearance. That is, they're typically attached to the ground or some other substrate. They'll have a stem or a stalk, and they have some kind of reproductive bodies. They come in a lot of different forms, but again, they are not plants. They're fungi, separate kingdom. The science or study of fungi is mycology, and the root word to this is myces, and myces means mushroom. What this means is that the first people that were looking at mushrooms, or the first people studying fungi, were looking at the macroscopic forms, the mushrooms. And it was important that they did because sometimes you can eat fungi. They're not super nutritious, but they do have some positive attributes in terms of being a food source. Sometimes people would eat them and they did funny things to their head. So they kind of wanted to know which ones can I eat and get that experience. And then sometimes people would eat them and they'd destroy their liver and they'd die. So you needed to know which ones were which. And that was kind of why people started looking at fungi and trying to understand which ones are safe to eat and which ones are not. At this point, we can categorize fungi into these three basic groups based on morphology. It's just handy. They are yeasts, molds, or fleshy fungi. The yeasts are typically single-celled organisms, so they're kind of like bacteria. Their growth is very similar to bacteria on a plate, but their cells are bigger because they're eukaryotic. And then the molds are filamentous and microscopic, and the fleshy fungi are made of filaments, but they're macroscopic. Some of them are quite large. We're primarily interested in the yeast types and the fungi, the mold type fungi. This is showing Rhodotorula glutinus. This is a type of yeast that is really common in wet environments. So if you have a bathtub and you don't clean it regularly and you start to see this pinkness developing somewhere near the drain or maybe up on the surface or around your sink, people think that it's Serratia marcescens because they're pink bacteria, but more commonly than not, when we've isolated pink organisms from sinks and so forth in here, it's Rhodotorula. So it's a yeast. And those guys, they're kind of hard to get rid of. They just keep coming back. But you'll notice these colonies, they look like bacterial colonies. Circular, entire, low convex, smooth, shiny, opaque. Yeah. So if you have a semester project that's pink, make sure that when you do your staining, you do an indirect stain first. Determine, is it really prokaryotic or is it eukaryotic? Eukaryotic cells are significantly bigger. If it's more than probably 2 microns in diameter, you get something that's up around 5 or 6 microns in diameter, it's probably yeast, especially if they're budding. And it's important for identification because we have a different primer set for fungi than we have for bacteria, for the ribosomal RNA. Anyway, another reason that fungi are not plants, fungi don't have true roots, stems, and they never have leaves. Those are characteristics of plants. Instead, fungi have what's called a body, or sometimes a term that might be applied is soma. Soma means body. But the body of a fungus is often referred to as a mycelium. And a mycelium is a mass of hyphae. But a mycelium is visible to the naked eye. If it's a mold-type fungus, you've seen mold-type fungi. A lot of you have had them on air plates, where the colonies that are produced are kind of fuzzy. Sometimes you see quite a lot of filamentous material sticking up into the air. If you're looking at a macroscopic fungus, one of the fleshy fungi, they're still made of threads. It's still a mycelium. The threads that make up the mycelium are called hyphae. So here we see a mycelium on the left, and those are fleshy fungi, although they're not very big. And then on the right, we see the individual hyphae. Again, hyphae are threads. Also, the word hyphae is plural. It's got an E on the end of it. If there's only one thread, it's a hypha. You almost never see one thread. But the mycelium, that's a fleshy fungus mycelium. If it was a mold-type fungus, it would look more like this. Oftentimes, a mycelium might just look like kind of a fuzz on the surface of something. The mold-type fungi are microscopic. The fleshy fungi are macroscopic. But they're still made up of hyphae. So hyphae, what are these things like? Again, these are thread-like filaments. They're typically microscopic. Sometimes they're big enough to be clearly visible to the naked eye. And last semester, a student brought in a fungus where the hyphae themselves, I hate to estimate how big they were. It's like, well, probably at least 1 millimeter in diameter, which is 1,000 microns. That's pretty good. So they were definitely visible to the naked eye. These thread-like filaments come together, the filaments intertwine, and they form this mass that is the mycelium. So hyphae may or may not be visible to the naked eye. The mycelium will be visible to the naked eye. And a mycelium typically has 2 parts. There's a vegetative mycelium, which is involved in food getting, and there is an aerial mycelium, which is involved in reproduction. If you're looking at fleshy fungi, usually what you see is just the aerial mycelium. And the vegetative mycelium is either in the ground or in a piece of rotting wood, or sometimes people find them growing up out of a wet carpet, which is not good. You don't want to find those there. But this is showing the whole mycelium. So the aerial mycelium is up on top, and the vegetative mycelium is down in the substrate, whatever it is the fungus is using for food. Now, up at the top of this says a fungus or the fungus cells form a true syncytium. Anybody know what a syncytium is?
A multinucleated mass of protoplasm?
A multinucleated mass of protoplasm. So there are some syncytia within our own bodies. Fungi form, the cells of a fungus form a syncytium. This is really important when you look at the structure of the mycelium because only the vegetative mycelium is exposed to the nutrients. The nutrients are down here. This is going to support the growth of that, which is going to come up and release a whole bunch of reproductive structures. It's really easy for nutrient to be passed from the vegetative mycelium to the aerial mycelium because the cells are all connected. They really do form a syncytium. By the way, that word syncytium is sometimes spelled S-Y-N and sometimes spelled C-Y-N. Don't ask me why. Anyway, nutrients are easily passed from one part to the next. These hyphae that make up the mycelium, again, we're back to the hyphae, they're the threads, they can be aseptate. What does A in front of a word mean? Without. And what's a septum? We have one in our notes.
Separating.
It's like a wall that separates. So if hyphae are aseptate, there's no cross walls. So if you look at them, actually you can't really see their nuclei here unless they're stained in a particular way. But if you were looking at aseptate hypha, this is multiple cells and they're not separated from one another by a cross wall. If the hypha is septate, then there will be cross walls and they look like this. But I just said those cells formed a true syncytium, which means the protoplasm is connected. It turns out that if you look at each septum, it's really not complete. It's like a lifesaver. There's a hole in the middle of it, so that there's exchange of protoplasm, nutrients, and so forth from cell to cell. It's always connected. They look like they form a complete wall across there, but they don't. Now, fungi are typically haploid. How many people are familiar with the term haploid? It means you have one set of chromosomes. So typically fungi cells have only one set of chromosomes. They're haploid. There are some fungi that have two nuclei that don't fuse. They don't really form a diploid zygote. They just have two nuclei. If they have that, they have two sets of genetic material, so they are referred to as being dikaryotic or dikaryon. What does the root word karyon mean? As in eukaryotic, prokaryotic? Nucleus. So dikaryon means two nuclei. So there are a whole bunch of fungi that have this situation where there's two nuclei per cell. And presumably they'd have to be septate in order for them, otherwise you couldn't tell whether the nuclei were in the same cell. Now, sometimes if you get into the study of fungi, you can get into the study of all the different names that apply to the hyphae. So I'm just going to show you a couple of examples. There are structures that are reproductive structures, conidiospores and sporangiospores, that we'll talk about in just a little bit. The hypha that supports those spores is called a conidiophore or a sporangiophore. So again, this is just the specialized name for this thread that is supporting a mass of reproductive cells. These are conidiospores. If the spores are in a sac, they're sporangiospores. But this is a conidiophore, and this is a sporangiophore. Not something I'm going to ask you about on the lab exam, although they're pretty easy to see. We have enough other stuff to look at. Some other specialized hyphae. There are hyphae called haustoria. This term doesn't apply only to fungi, because if you read about mistletoe, which is a type of plant parasite, they also form haustoria. Haustoria are just extensions of the protoplasm that penetrate into a host cell. The root word here, haust, means to suck. So there's an illustration showing haustoria. They're extensions of hyphae that actually go into the host cell. So here you see the fungus filaments, the hyphae, and they go down into, this is a plant cell, so they're going in. Here you can see actual images. These plant cells, each one of them has a haustorium. There's only one, so each has one haustorium going in. Haustoria then are simply going to absorb nutrients from the host, so they're produced by fungi that are parasites. There's another type of specialized hyphae called mycorrhizae. Mycorrhizae are produced by fungi that are not strictly parasitic. In fact, they're usually thought of as being mutualistic. So we had different groups or different categories of symbiosis. If the relationship is mutualistic, then both organisms in the relationship benefit. So the fungi that produce mycorrhizae, these are associated with plant roots, and sometimes actually root hairs, which is kind of hard to tell in this image. But this is an electron photomicrograph. This is a root hair, and these threads are mycorrhizae. And what they do is extend the plant root system so that the plant has more fibers going out into the soil. And they can be ectomycorrhizae, where they don't actually penetrate the root, or they can be endomycorrhizae, in which case they do. But they're not considered parasitic. As soon as I say that, and I think, well, they're not considered parasitic, but I'm pretty sure the fungus benefits by getting nutrient from the plant, because plants convert carbon dioxide into sugar, and they transport that sugar down to their roots so their roots can grow. They also help the fungus grow. Meanwhile, what the fungus is doing by extending the plant root system out like that, they're going to help that plant to get water and minerals from soil that they wouldn't otherwise be able to get. The mycorrhizae then are very important to the growth of lots of different types of plants, particularly forest trees. And in certain parts of the country, this country and other parts of the world, people are finding out where humans are harvesting too much fungi. So if humans are picking all the fungi, there's not enough fungi to support the trees because the mycorrhizae are essential to the trees. So if you are a harvester of wild fungi, reconsider. Anyway, this is going now into fungus reproduction. When we think about reproduction of eukaryotic organisms, typically we think, well, they can all probably reproduce both sexually and asexually. With fungi, there was a long period of time where people thought they could not reproduce sexually. So they had this category for fungi that couldn't reproduce sexually. They called them the fungi imperfecti, imperfect fungi. They don't know how to reproduce sexually. And then it turned out after time, it wasn't that they didn't know how, it was that the conditions weren't right. And they're not going to reproduce sexually if you don't have soft music, low light, alcohol. Don't believe everything I tell you in here. But it turns out fungi actually can reproduce, they much more commonly reproduce asexually than sexually. Can you think of a reason why they might? Think bacteria for a moment. Bacteria reproduce over and over and over again. Transplant them to a plate, transplant them to a plate. They're doing that asexually. Why is it easier than sexual reproduction? Sexual reproduction, if you think back to our definition, requires a reorganization of the genetic material, which means you have to have a source of different genetic material. So from a fungus standpoint, you know, you can reproduce all by yourself. It's way easier than having to go out, find another fungus, maybe buy them dinner, go through that. Anyway, they reproduce asexually much more commonly. It's just easier. Under experimental conditions or the artificial conditions that we grow fungi, what researchers found out is they had to give the fungi a specific type of medium. Probably had to do with certain minerals or vitamins. If they didn't put them on the right medium, they would not reproduce sexually. They also had to have two genetically different fungi. And the fungi could tell. You put them on there with the one that's exactly the same genetically, they don't reproduce sexually. You're gonna have somebody with some variation. Okay. Asexual reproduction is always going to involve growth of the cell. They've got to get bigger, and then they have to undergo mitosis, which is a process involving the separation of chromosomes. And because this is a microbiology class, I'm not going to talk about mitosis. It's like you got to have all these different phases, and never mind. But separation of the chromosomes. Anyway, we say that when fungi reproduce and one cell divides in half and forms two cells, that's called binary fission. Binary fission is across the long axis. So fungi typically form cells of hyphae that are somewhat linear, kind of like a rod-shaped bacterium. When fission occurs that's binary, it goes across the long axis. One cell just becomes two. So here we see one fungus cell, looking kind of like a box. But that one fungus cell just divides into two fungus cells. The division across the long axis is binary fission. Some fungi reproduce by budding. And budding is a little bit different because although it still involves mitosis, separation of chromosomes, the cytoplasm is not divided equally. Instead, half of the nuclear material goes into a bud, and the bud just gets a tiny little bit of protoplasm, and it's like this little piece that's kind of sticking on the outside of the original cell. In a way, it helps the fungus ensure its survival. This is really common for yeast, so if you have one yeast cell, it'll produce a bud. If the bud grows up, it'll come off and be a new yeast cell. But if conditions get bad and it can't survive, that bud will just fall off, and the original cell hasn't lost much. So here we have a cell, here we have a cell with a bud. The nuclear material amount is the same, but the cytoplasm around it is really small. Sometimes you can see yeast cells that have more than one bud, and when the buds fall off, they leave bud scars, these little spaces on the surface of the cell. And then another form of asexual reproduction is fragmentation. Well, if hyphae are septate, each one of those separate segments can be broken off and can potentially grow a brand new fungus. So fragmentation is just breaking up a hypha. And then they can also reproduce asexually by forming spores. If you were actually taking a mycology class, you would find out that there's a lot of overlap in terminology, and although it looks like I can show you these 4 very distinct ways that the fungi can reproduce, when you start looking at the reproductive structures, they're almost always called spores. It's like, wait, wouldn't they be called buds and fragments? Yeah, but they're also called spores. So when you have a fungus that reproduces with bud-like structures, those are sometimes called blastospores. And if they make fragment-type structures, those are called chlamydospores. So here we see some examples. This is a fungus. You can see these little pieces that come off. There's a little bud. Here's a much bigger bud. A little tiny bud. Those things can come off. For whatever reason, those are called blastospores, even though they are buds. This is the same kind of thing. These are growing off to the side. They start out small and then they get bigger. So those are buds, but they're also called blastospores. And then these are chlamydospores. And chlamydospores are just fragments. So you can see where there was a hypha. Chlamydospores typically have thicker walls than the rest of the fungi cells in the hypha. But anyway, they're fragments. By the way, those terms, blastospore and chlamydospore, I'm not going to ask you those names on the test either. That's getting into too much detail about fungi. These, on the other hand, you need to know. So these we'll be looking at in the laboratory. These are 2 types of asexual spores that you will be responsible for. Conidiospores and sporangiospores, which are shown in the next slide. What's handy here is conidiospores. We've already seen that term. Conidiospores were the structures made by filamentous bacteria, Streptomyces, that come off the end of the filament. It's not too surprising the terminology is the same because people used to think Streptomyces were fungi. And that was before they realized the difference between eukaryotic and prokaryotic cells. Now they know Streptomyces are prokaryotic. They're not fungi, but the terminology stays the same. So conidiospores, we have 2 examples here. This is Penicillium. Penicillium, the conidiospores are arranged like beads on a string, where they're more or less in line with one another, kind of like the fingers on your hand or the straws of a broom. So they're parallel. This is Aspergillus, and here the conidiospore chains are coming out in all directions from a central point, so it's more like a dandelion puff or a koosh ball. You still have koosh balls? Maybe. Anyway, the Greek word conus is dust, and that's where the term conidia comes from. These things, when you look at the surface of a fungus, and there's a couple of people that have fungus colonies that have these little dark spots all over the top that are really distinctly visible to the naked eye. Those little dark spots are these things. They're just loaded with spores. And if you look at the surface of an orange, for example, very often if you find an orange that has fungus growing on it, which you usually find it inside the bag, if you buy a bag of oranges, it'll be in the middle, someplace hidden, and you pull it out and it's got blue or greenish spots on it. That's conidiospores, usually these guys. And again, if you touch it, that stuff comes off on your fingers, and then you've got that blue-green stuff, or you put it down someplace, it falls out. Dust, it's conidiospores. And every single one of those can grow a new fungus. So these guys have tremendous reproductive potential. These are sporangiospores. Sporangiospores are in a sac. In this case, the sporangium is linear, but they're not always. So this is a linear type sporangium, but sporangia are often round. So this is an example of a sporangium that's more circular or spherical. Anyway, a sporangium is a sac. By the way, the term sporangium is also the name of a cell that contains an endospore. A lot of terminology similarities because bacteria used to be considered plants also. Back in the beginning, there were only animalian plantae. So fungi were plants, bacteria were plants. What do we know about archaea? Okay. Now, when we're looking at sexual reproduction, as I said, it's a little more difficult because the fungi have to get together with a fungus that's genetically different. Sexual reproduction is going to involve 3 stages or steps, and these are called plasmogamy, karyogamy, and meiosis. Plasmogamy, GAM or GAMO, GAM, either GAM or GAMO, means union or marriage. And the plasm has to do with the protoplasm. So plasmogamy is the union, marriage, or joining of the protoplasm. So if we have 2 hyphae, and they have to be genetically a little bit distinct from one another. As soon as I say that, I think, but really fungi don't have male versus female in the usual sense because they don't have sexual dimorphism. And so usually what researchers will refer to them as the plus strain and the minus strain if they're genetically different. Anyway, so they have to recognize that they're different, and then they're going to join their protoplasm, like so. That's plasmogamy. So where you had 2 separate cells, the wall disappears between them, and now the protoplasm has been joined, and we're beginning, that's the first stage of sexual reproduction. Let them meet and join up their protoplasm. The next step is karyogamy. And again, gamos means marriage or union, and karyon means nucleus. So this is going to be the joining of the nuclei. So here we see our nuclei separate, and then we're going to bring our nuclei together, and they're going to fuse. When they fuse, they form what's called a diploid zygote. That's the term that usually applies, but technically, that thing isn't really diploid. Diploid means you have 2 sets of chromosomes. At that point, it actually has 4 sets. It's tetraploid. But it's because the chromosomes, when you look at a chromosome, it's an X-shaped structure. That X is 2 sets of DNA. If you're just counting the chromosome, if you count each X as one type, then it is diploid. Anyway, so we've got our nuclei joined up, and then the next step is meiosis. Meiosis is sometimes referred to as reduction division. What's going to happen initially is the chromosomes line up kind of in the middle of the cell, but they line up in homologous pairs. In this illustration, we have very large chromosomes, little bitty chromosomes, and medium-sized chromosomes. The 2 large ones are together, the 2 little ones, and the 2 medium-sized, so they're lining up in homologous pairs. Homologous means they're similar, they have genes encoding the same characteristic in general, but they're not identical. So one chromosome comes from one partner, say the minus strain fungus, one comes from the other, the plus strain. They line up in pairs, and then eventually those pairs are going to separate, but while they're lined up like this, the fungi engage in what's called crossing over. And here's a diagrammatic representation. Those X's are not just sitting near one another, the DNA begins to entwine. And it entwines a lot, and then they exchange pieces. So you've got one set of genetic material from one parent, one from the other. When they get together during meiosis, they exchange parts. So there's a tremendous amount of genetic variation introduced here. This is showing red and blue combination, but it's more complex than that. There's a lot of it. Eventually, the chromosomes are going to separate into our daughter cells, and with meiosis, there's actually 2 sets of cell divisions. First time the pairs separate, and then each chromosome separates into 2 chromatids. So another thing that happens during meiosis, you take a cell from what was diploid initially back to haploid. Haploid means only 1 set of chromosomes. And the offspring are going to be what we call the sexual spores of fungi. We're going to be looking at these in the lab. They are oospores, zygospores, ascospores, or basidiospores. And that has to do with their arrangement and so forth. But what you'll see in this illustration, if this was the original zygote, you've got all these variations on a theme, different arrangements, and that's not counting crossing over. So just separating the chromosomes and then separating again gives you variation, but crossing over gives you a lot more. So the most important aspect of sexual reproduction is it allows for genetic recombination. It allows the offspring organisms to be distinctly different from the adults, and that gives you variation in your population. So if conditions become bad in the environment, some fungi will survive. They can't all survive, but some will because you've got all this variation in the population. Now, this is going into some significant features of fungi. One, they're saprotrophs. What does that mean again? They are decomposers, so they get their nutrient from dead, decaying material. Imagine, if you would, for just a moment, that you lived in a world where there was no decomposition of organic material. Have you ever been to the woods in the fall, and you're walking along and there's all these leaves on the ground? Imagine about 100 years worth of leaves piled up. You wouldn't be able to go anywhere. Anything that fell on the ground, instead of decomposing, it would just sit there. And pretty soon there would be layer upon layer upon layer of dead stuff piled all over. So saprotrophs really are important because they recycle. They break that material down and put the organic material back in the soil. Another feature, some fungi form mycorrhizae. So I just talked about the mycorrhizae. These are extensions of the roots. They're fungi that they make these extensions of the roots of plants, so they're actually going to help the plants get water and minerals. When the mycorrhizae are missing, well, oftentimes forest trees can't get the water they need, and they die. One of the primary minerals that they pick up is phosphorus, and phosphorus is essential for making ATP and for making nucleic acids. And then some fungi are a food source. How many of you eat fungi? Be honest now. On your pizza, mushrooms. Stuffed mushrooms.
Mushrooms.
In your salad, mushrooms. If you've ever eaten brie, that white fuzz on the surface of brie cheese, that's mycelium. That's fungi. You're eating the cheese, but the fungi were eating it first. Fungi are also used in food processing. Typically, we think about them being associated with beer production because Saccharomyces cerevisiae, the name basically means sugar fungus that makes beer, cerveza. Anyway, Saccharomyces cerevisiae is a fungus, the yeast. They're also involved in bread production, bread that is fluffy. The air, the fluff, was actually carbon dioxide, metabolic waste product. And then some fungi produce antibiotics. Which one do we know that produces antibiotics that Alex Fleming found? Penicillin. So penicillium is the fungus that produces penicillin. There are other fungi that produce other types of antibiotics. Fungi are being used industrially now. A lot of times people were looking at fungi and trying to find different enzymes that fungi could produce, using them a lot of the ways similar to what enzymes from bacteria could be used for. But now what's happened because of genetic engineering, Saccharomyces cerevisiae is being used to produce all kinds of things. Basically in laboratories, somebody says, I want my Saccharomyces to do this. Somebody just puts the genes in there and says, here, you do that now. So they produce enzymes that are used. They can also make organic acids, solvents. There's a type of fungi that has been discovered, and I think it was discovered because somebody was out in the woods and began to smell gasoline. And they couldn't figure out where the smell was coming from, and then they found a fungus that was producing a substance that was about the equivalent of jet fuel. And then, as I said, recombinant proteins, they can produce a wide range of things where genes are taken out of other organisms and put into fungi, and then they will produce the proteins of that other organism. Some fungi form hallucinogenic substances, which is good or bad depending on how you look at it. And then some fungi are potentially pathogenic. So this is generally not considered beneficial, but definitely a significant aspect of fungi. And this leads us into our next general topic, which is the study of medical mycoses. The term mycosis means disease caused by fungus. As far as I know, it only applies to humans and animals, though. I think when plants are infected by fungi, I've never heard the term mycosis apply. Not sure why. But anyway, mycoses is plural, mycosis is singular. Again, fungus-induced disease, or a disease caused by fungi. Kind of an interesting aspect, there are actually more fungal-induced diseases in humans currently than there used to be. Now, part of this might have to do with diagnostics. It's easier for people to recognize fungal infections, but there are three factors that are recognized as increasing the incidence of fungal infections in humans. One is long-term use of antibiotics. Remember, we have normal microbiota, and our normal microbiota is both bacteria and fungi. They are normally with us. If you start killing off the bacteria, which is what antibiotics do, the fungi then can overgrow. They've lost the competition. It allows the fungi to overgrow and oftentimes begin to cause disease symptoms. We have Candida that lives with us. It's part of our normal microbiota. Most people do, different strains, and Candida can be a disease-causing agent. So the fungi can begin to grow out of control, and then they cause disease. Another factor that has increased fungal infections was the use of chemotherapy for cancer patients and for transplant patients. Basically, chemotherapy for these types of situations knocks out the immune system. Fortunately, now there are some really good drugs that can be given to increase white blood cell count, so bring your white blood cells back up to a normal level. But chemotherapeutic agents can be quite devastating. And the third reason was infection with HIV. HIV infection, that virus just wipes out the immune system. We'll talk about this in quite a bit of detail later on. So, long-term use of antibiotics, chemotherapeutic agents for transplants and cancer patients, HIV, what do they have in common? Knocking out your normal immune function. Your normal microbiota are critical to normal immune function. They assist you. They help prevent other organisms from growing. So basically, whether you get infected by a fungus or not has to do with the state of your immune system. With bacteria, you have specific species of bacteria that are much more virulent, where they produce some really nasty byproducts that can cause disease symptoms. Fungi don't seem to be like that. Fungi just grow. They take up nutrients. They are eukaryotic. They can compete with your cells very successfully. Whether they get to do that or not is dependent upon your immune system. If your immune system is functioning normally, you can keep them under control. If your immune system is not functioning normally, look out, because they're very likely to cause problems. So, that said, we're going to look at some categories of mycoses. Again, this is biology. We categorize everything. So they get put into categories. Sometimes there's 4 categories. I'm just going to use 3. The categories are superficial, subcutaneous, and deep or systemic. So we'll look at these 3 categories of mycoses, what's going to be happening. So superficial mycoses. These are fungal infections that involve fungi growing on the surface of the skin, or they can grow in association with hair follicles, and sometimes they do damage to the hair. They can get into nail beds and cause damage to the nail. Superficial mycoses are caused by a group of fungi that are often referred to as dermatophytes. Unfortunately, the term phyte means plant, and fungi aren't plants, but dermato has to do with skin. And as far as I know, that term is still used for this group. There are multiple genera: Trichophyton, Epidermophyton, Microsporum. These are 3 different genera of fungi that cause superficial mycoses. Here you can see some reproductive structures. These things produce, these are called macroconidia. Yeah, again, they look like fragments to me, but they also produce microconidia, these little things here, and these are more like buds. They have a tremendous reproductive potential. So some examples of superficial mycoses. One is Tinea pedis, which is more commonly referred to as athlete's foot. Now be careful here, because you see the terms on the left. Tinea pedis, 2-part name, the first one is capitalized. That looks like a genus and species name. It is not. That's the name of a disease. A mycosis is a disease. It's an infection of fungi. So Tinea pedis is athlete's foot. And you've probably seen the commercials, athlete's foot, where you see the foot and it's all red and flames are coming off of it. A lot of people have at least some experience with athlete's foot. Where the fungus gets in, it causes damage to the skin, it gets into the nails can crack and become quite painful. Oftentimes, secondary bacterial infection can get set in, and then it can be more problematic. The next one, Tinea capitis, this is ringworm of the scalp. the fungus is just under the surface of the skin, and right in the area where the fungus was growing, they cause inflammation, and so the skin would be red and a little bit elevated, and people thought there was a worm underneath. That's where the name ringworm comes from. If it's associated with the scalp, it often involves Trichophyton mentagrophytes, which is a fungus type that they make hyphae that actually penetrate into hair, and they will break the hairs off, and then you'll see bald patches occur if a person has ringworm of the scalp. And then Tinea corporis is ringworm of the body, same kind of thing, different species. But oftentimes the dermatophytes, you can have different ones that cause similar symptoms. They're not necessarily restricted to one. So here's athlete's foot in 2 images. This is ringworm. So this is Tinea corporis, but this ring, again, this is an elevated, kind of red, usually itchy, and that's due to inflammation where the fungus is actively growing. Think about this as like a bacterial colony. The fungus started growing here, they're growing outward, and so this is the region of most active metabolism. That's where you're going to see the most irritation, and that's why it tends to form a ring. These become much more irregular as this grows outward. And it's usually very itchy, irritating. Next category is subcutaneous mycoses. Subcutaneous mycoses involve fungi that have been introduced under the skin. So they're between the skin and the underlying muscle or underlying bone. Typically puncture wound, cut, bite, something. They have to be introduced. And if your immune system is functioning properly, they tend not to spread, so they're kind of localized. Hope that your immune system can control these things. One of the most common involves a fungus called Sporothrix schenkii. Sporothrix schenkii is often associated with vegetation where people are working in their garden, they get stuck with a thorn, rose thorns, berry thorns, that kind of thing. And then where the fungus is introduced, you can have an infection develop. This is what it looks like on a plate. This is what infection looks like. Often on the hands, sometimes on the feet if people are walking around barefooted, but often on the hands because people are handling plant material and they get stabbed. Another one, there's chromomycosis. Chromomycosis refers to infection where the fungus involved is dark colored. There are quite a number of them that have dark pigments in their hyphae, and then oftentimes you'll see these raised areas that are dark. Those are just called black molds. And then the last category are the deep or systemic mycoses. These, typically the fungi get in by means of the respiratory system, but not always, because sometimes they can get in through an accidental injury. The fungus basically gets into the bloodstream. Once they get into the circulatory system, they can go to internal organs. And again, if your immune system can't control them, they can cause considerable damage. One type of fungus that is typically associated with systemic mycosis is called Coccidioides immitis. These are fungi that live in the soil, but they tend to like a particular habitat. And so they, in California, they're associated with the lower part of the San Joaquin Valley. They're also found in the Four Corners region, or the Four Corners, California, Arizona, Utah. Anyway, where those, what's on the 3 years?
Utah, Colorado, Arizona.
Colorado? Okay, I was like, what's the 4th one? But anyway, it's desert. So these things like a particular type of soil, and it tends to be deserty. They grow in that soil, and they form these hyphae that then break into arthrospores. Again, arthrospores are fragments, kind of thick walls. It's really easy for those things to get picked up in the wind and then blow around, and people breathe them in, and that's how they get infected. So coccidioidomycosis is the technical name for infection with Coccidioides immitis. It's also referred to as San Joaquin Valley fever. Probably not if you live in the San Joaquin Valley because you don't want your home to be named that way. But it's also called desert rheumatism, or sometimes just valley fever. And if a person is exposed to Coccidioides immitis, most people that are exposed don't experience any symptoms whatsoever. Again, because most people have a fully functional immune system and the Coccidioides is kept in check. If they develop flu-like symptoms, that's when some trouble is brewing, because that means their immune system is not controlling the fungus, and it happens only in about maybe 10% of people that are exposed, maybe even less than that. But if you had been in an area where, if you were in the San Joaquin Valley and there was a dust storm, a lot of wind blowing around, and you develop flu-like symptoms, you might want to be paying attention to what else is going on, because the next thing that happens is you'll develop a skin rash. If you develop the flu-like symptoms and then the skin rash, that means coccidioides immitis is spreading throughout your body, and it can cause severe damage to internal organs. It can be fatal. So there's the skin rash. The coccidioides is spreading throughout the body. There is a test for coccidioides infection. We're going to be doing a test in lab later on that's referred to as the Auchterlonie test. Anyway, it's often used as a way of testing for antibodies against coccidioides. Another organism that causes deep or systemic mycosis is Histoplasma capsulatum. Histoplasma also is a soil fungus. They tend to like soil that has a very high nitrogen content. So in places where there's a lot of bird droppings or bat droppings, people that do spelunking, spelunkers lower themselves down into caverns and then creep around in these caverns, and if there's bats in there, sometimes they're coming face to face with bat droppings. Histoplasma typically causes pneumonia-like symptoms, so it gets into the lung. Again, systemic mycoses involve fungi that usually get in by way of the lungs. But then again, if your immune system can't control them, they can get throughout the body and cause considerable damage. And then it turns out that pretty much all fungi can be opportunistic pathogens. So there are certain ones that are recognized as being real common causes of mycoses. The ones that I'm going to mention next are less common, but still possibly can cause disease. Notice it says if the person is immunocompromised. So if your immune system is really damaged, any fungus is potentially pathogenic, even Saccharomyces cerevisiae. And that's purchasable in a supermarket. Yeast. Well, okay, one of the most common causes is Candida. Candida albicans is, again, part of our normal microbiota. Most of us have Candida. They live in the mouth. Your female, they live in the vagina. They live in the GI tract. If your bacteria that normally keep Candida in check are reduced significantly in number, Candida can get too populous and they cause infection. They can cause vaginal infections. They can cause thrush infection in the mouth. If they get into the bloodstream and your immune system can't control them, they can cause septicemia, which is potentially fatal. So here we see Candida. Again, these are yeast-like fungi. Here you can see the bud scars on the surface of these yeast cells. This is what it looks like on a plate. Here we see what are called pseudohyphae. So under certain circumstances, Candida can begin to form these things that look like hyphae, even though they're considered a yeast-like fungus. And I should say, that's not really uncommon for fungi. There are quite a number of fungi that grow like yeast under certain conditions and grow like multifungi under other conditions. There's a new type of Candida called Candida auris. This is just being detected recently, and apparently this one is really hazardous because it's really difficult to control and very rapidly spread. So it's showing up in clinical settings, and no matter what people are doing to try to control this thing, they tend to get all over the place. They can get into the bloodstream, cause septicemia, cause quite invasive infection. And as I said, they tend to persist even though people are trying to control them in the clinical setting. They tend to persist and cause problems. So this is it's a newcomer, but it's quite a worry. These are drug resistant too, which makes them more hazardous. Then there's Cryptococcus. Cryptococcus neoformans is a positive agent of a pneumonia. In HIV patients particularly, these things were associated with bird droppings, and a lot of people are exposed to bird droppings in cities because there's pigeons and whatnot. They roost on window ledges, they're in the parks, they're walking all over the place, and so people would be exposed to Cryptococcus. Unfortunately, these guys can get into the lung, and then they get into the bloodstream. They tend to have an affinity for brain tissue, and they cause meningitis, which is potentially fatal. So there we see Cryptococcus neoformans colonies on a plate, and the culprit spreads them standing on a park bench. There's another Cryptococcus called Cryptococcus gattii. This is supposed to be restricted to the tropics, but they've shown up recently as far north as Seattle, up in Washington, and in the forests up there. They're apparently associated with trees. It was thought that they were associated with just certain trees, and then I guess a group in Davis found them associated with lots of different kinds of trees, including the eucalyptus that are all over the place. These can cause soft tissue infection, lung infection, joint damage, meningitis. So there's Cryptococcus gattii. Then there's Aspergillus. There are quite a number of species of Aspergillus that are potentially disease-causing agents. So Aspergillus fumigatus, they produce conidiospores that are very, very tiny, and so it's easy to encounter large numbers of these things. If you breathe them in, typically they cause pneumonia-like symptoms. If they get introduced into the bloodstream, say a lawnmower accident or something like that, they get into the bloodstream, if your immune system isn't working properly, these guys can cause septicemia and kill you fairly rapidly. Fungi grow very quickly under the right conditions. Even Rhizopus stolonifera. Now, this thing is usually referred to as bread mold, really common fungus, not usually pathogenic. They can get into the lung and cause pneumonia-like symptoms. This is what Rhizopus looks like on our prepared slide. These are the sexual spores of Rhizopus. These are the asexual sporangia of Rhizopus. But if they get into tissue, then they look something like this, where you've got hyphae extending throughout lung tissue, and they are potentially very damaging. Again, they're eukaryotic, they take nutrients that you want to use, and if they're in the lung, they can do a lot of damage in there. They form fungus bones. Not good. Pneumocystis carinii, this one used to be thought of as protozoa. It took a while for people to figure out they're not protozoa, they're fungi. Again, they cause pneumonia-like symptoms, typically in immunocompromised individuals. There's pneumocystis. I think I'm going to stop. We're getting into the section about mycotoxins. So this is a good place to break. And then, so if you're studying, when you're looking at the questions in the study guide, pay attention to not questions about mycotoxins, because we're not going to do mycotoxins on next week's quiz. For the bacteria, we have all those different genus names. Expect to see a matching type situation. Maybe we'll have two matching situations. I don't know. Anyway, it's a lot to cover, but I said that at the beginning, didn't I? We go through a lot of material in this class.